Bacteria resistant to antibiotic are currently responsible for more than 700,000 human deaths each year. They could lead to the deaths of 10 million people per year globally by 2050. Bugs be gone. Superbugs is bacteria resistant to antibiotic currently in use are on the rise. Liz Sheffield describes two new weapons in our armory. Okay, now we are going to bugs be gone. Bacteria resistant to antibiotic are currently responsible for more than 700,000 human deaths each year. They could lead to the deaths of 10 million people per year globally by 2050, according to a recent report commissioned by the UK government. This estimate is based on the antibiotic currently approved for use to treat disease, which of well, most of which have been in service for more than 50 years. During that time, their use in human and other animal has exerted selective pressure on bacterial population, favoring the survival of those with genes conferring resistance. After more than 30 years during which no new classes of antibiotic were discovered, 2015 saw one new class identified, and this year has already seen two new breakthroughs in the fight against superbugs. Thank you. Now for the nanobug trap.
Okay, thank you, Bara. If harmful bacteria enter the bloodstream, they can multiply so rapidly as to cause threatening conditions including organ failure and sepsis. There are two ways to treat infected patients. One is to administer antibiotic. The other is to pass their infected blood through a filter in dialysis machine. This second method is the only option with antibiotic-resistant infections, but has until now relied on carbon foam-based filters, which capture only 10% of the bacteria in the blood. Now researchers in China, inspired by the famous fly trap, have devised a claw-like filter that grabs 97% of the bacteria passing through. Okay, Albert.
Okay, thank you, Vito. Now for my report. The claws of the filter are made from flexible polycrystalline nanowires type with concanavalin A. This protein binds to certain carbohydrates, including the sugar that coat the surface of bacteria such as Salmonella. The nanowires stick to and close over the bacteria, trapping them in a delicate cage and preventing them from re-entering the patient's bloodstream when the blood returns to the patient.
All right. Thank you, Albert and Alexander, for the news on nanobug trap. Now, for new antibiotic unearthed. Albert?
Okay, thank you, Bara. So most of our best known antibiotics were originally obtained from fungi or bacteria cultivated in laboratories. The most renowned example is penicillin, which was derived from a fungus. We know that there must be many more antimicrobial compounds produced by microorganisms in the natural environment, but finding conditions and media suitable for cultivating them has proved challenging. Molecular biology techniques now allow researchers to avoid the need to cultivate organisms and go straight for the genes that carry the information necessary to make compounds of interest. Now for Hizkia.
Okay, thank you, Albert. A team in the USA recently extracted DNA from 2,000 different soils and used the polymerase chain reaction to amplify DNA sequence known to be associated with the production of bacteria while boosting compound. They found a sample from a desert region that filled the bill. They cloned the relevant genes and rearranged them to insert into the host bacterium Streptomyces, which grew reality in the laboratory. The bacteria made a novel compound, which the research called malacidin, from Latin words for killing the bat, and which work differently from any current antimicrobials. It has already been used to clear infection from rats with the superbugs methicillin-resistant Staphylococcus aureus or MRSA. Even better news is that after 20 days of continuous exposure, MRSA bacteria showed no signs of developing resistance to the new antibiotic. Thank you.
Okay, thank you, Hizkia. Thank you, Albert, for the news. Thank you, Alexander, too, for this news. And thank you, people, for watching us. See you next time. Bye bye.